fats and oils. Today I am going to explain about the topics including lipids, simple lipids, triglycerides, fats and oils, identification fats and oils, hydrolysis and rancidity. Let's sneak a look through the topic of lipids. Lipids are a group of heterogeneous compounds related by their physical properties rather than their chemical properties. They are activated in water and soluble in polar solvents like benzene, ether, chloroform, etc. Their major functions includes they can be stored form of energy and they provide insulation and give shape and contour to the body. Protect the internal organs by cushioning effect. It helps in absorption of fat-soluble vitamins like A, D, E and K. Lipids are classified into three major groups. They are simple, complex and derived lipids. Again, simple lipids are divided into fats or oils and waxes. Complex lipids are classified into phospholipids, glycolipids, sulfolipids and lipoproteins. And derived lipids are fatty acids and alcohol. Let's get a detailed view about the simple lipids. Simple lipids are esters of fatty acids and alcohols. They are classified into two classes, fats or oils and waxes. They are uncharged lipids. Fats in the liquid state are known as oils. Waxes are esters of long-chain fatty acids and long-chain monohydric alcohol. For example, triacylglycerol, cholesterol esters. Now we can talk about one of the most important neural lipid in our body, triglycerides, as an example of simple lipids. Fats and oils are triglycerides stored by our body. They are esters composed of three molecules of fatty acids combined with the glycerol. They come from the food that we eat as well as from being produced by the body. Here, three fatty acid, the equation. Here the three fatty acid is combined with glycerol and it is reacted in the presence of a catalyst and triglyceride is formed. Our next topic is about fats and oils by differentiating it. Solid fats are tags with more saturated fatty acids and exist as solid in room temperature with high melting point. Whereas oils are tags with more unsaturated fatty acids and exist as liquid in the room temperature with low melting point. Fats are saturated fatty acids which have crystallization stabilized by the hydrophobic interactions, whereas oils are three cis double bonds of the unsaturated fatty acids introduce bonds in the hydrocarbon tall and inhibit the close packing. The source of fat is usually from animals and in case of oil, it comes from plant sources. Some of the examples of fats are butter, ghee, dalda, etc. And example for oil is sunflower oil, groundnut oil, etc. Next, we are going to discuss about what fats and oils are necessary to identify. To know the purity of the fat, to assess the degree of adulteration, to determine the proportion of different types in a mixture, to identify different types of fatty acids and degree of unsaturation. Now, fats and oils do have their reactions. Here, we are going to discuss about two major reactions. First one is hydrolysis. Triglycerides are easily hydrolyzed by enzymes called lipases in the digestive tract of animals to give fatty acids and glycerols. The fatty acids so produced play an important role in the metabolic process in the animal body. Here, in the reaction of hydrolysis, triglyceride is hydrolyzed by the enzyme to give glycerol and three fatty acids. Now, another reaction of fat and oil is rancidity. Rancidity is the process of oxidation of fatty acids by which fat produces objectionable odor and color. Rancidity in lipids may be due to hydrolysis or oxidation. The rancid fat is unpleasant to taste and smell. It can be measured by acid number. Rancidity and lipid peroxidation can be prevented by adding antioxidants like butylated hydroxyanisole and butylated hydroxytoluene as food additives. That is, it is used to prevent the spoilage of food. Rancid oils and fats are not suitable for human consumption as they affect the integrity of biomembranes, which will lead to cell death. In simple words, we can say that rancidity is the spoilage of food. Now, rancidity are of two types, hydraulic and oxidative rancidity. In hydraulic rancidity, this occurs when the fat breaks down by hydrolysis reacting using the water present in the body. Whereas in oxidative rancidity, this occurs when the unsaturated fat reacts with oxygen from the air. The site for hydraulic rancidity is the ester linkages in the triglycerides, and the site for oxidative rancidity is carbon double bond carbon in unsaturated triglycerides. In hydrolytic rancidity, it occurs more readily in the presence of heat, so refrigeration can be used to reduce the process. In oxidative rancidity, it occurs more readily by light, enzymes or metal ions, so it can be reduced by adding antioxidants. Therefore, I am concluding my topic by saying that lipids play a much more important role in the human body and they are considered as essential and need to be supplemented in our diet of our habitual life.